Okay, so good evening, you all. My name is Albert Prasad, and I'll be discussing on the Plasmodium species. So, to start off, there are four different types of Plasmodium species. The four types include Plasmodium falciparum, Plasmodium malariae, Plasmodium ovale, and Plasmodium vivax. So, we look at the life cycles, the pathogenesis, the etiology, the preventions, and the treatments available. So let's start off. Introduction. So there are approximately 156 named species of Plasmodium, which infect various vertebrates. So these Plasmodium species, they are the ones responsible for malaria, which is a significant global health issue. The primary species causing malaria in humans are the four types, right? Which I have already discussed. So let's learn about what disease it causes. So first is Plasmodium falciparum, causes the most severe and deadly form of malaria. Second, Plasmodium vivax, known for causing recurring malaria due to its ability to form dormant liver stages called hypnozoites. Third is Plasmodium ovale, similar to Plasmodium vivax, but it can cause relapses due to hypnozoites. And the last one, Plasmodium malaria or malariae, causes a more chronic and less severe form of malaria. Chronic meaning that it will last for like many years. Let's look at each of them. So Plasmodium falciparum. So they are predominantly found in sub-Saharan Africa, but also present in Southeast Asia, South America, and other tropical and subtropical regions. And they are responsible for the majority of the malaria-related issues worldwide, which leads to severe cases of deaths. And they are transmitted by the Anopheles gambiae, a complex of mosquitoes which is found in Africa. Second is Plasmodium vivax. So they are widely found across Africa, prevalent in Asia, Latin America, and parts of the Middle East. So they cause less severe, so they are less severe than the Plasmodium falciparum, but as you know that they can cause relapses due to dormant liver stages, which is called the hypnozoites. Transmission, they are transmitted by the Anopheles species, including the Anopheles stephensi and Anopheles albimanus. Moving on, third is Plasmodium malariae. So they are found in sub-Saharan Africa, parts of Southeast Asia, the Amazon basin, and the Western Pacific. So they cause chronic infections, a long-lasting low-level parasitemia, which is less severe but can lead to nephronic syndrome. This transmission, it is transmitted by a variety of Anopheles species, referring to the mosquitoes, right? The different varieties of mosquitoes. So the Anopheles is one variety that has this Plasmodium malariae carrying with them, which cause the disease malaria. Last one, Plasmodium ovale. So found in West Africa, but also in Central Africa, the Philippines, Indonesia, and Papua New Guinea. So they cause relatively mild malaria with potential relapses due to hypnozoites. Transmitted by Anopheles mosquitoes, particularly found in forests. Now looking at their pathogenesis. Okay, so the pathogenesis and the life cycle, they are similar. They are actually the same, but in a pathogenesis, so I'll just go through the points in pathogenesis, but in life cycle, we have like diagrams for you to understand the each points that I'll be discussing in these steps. So first is a mosquito vector. The start begins when an infected female Anopheles mosquito takes a blood meal and injects the sporozoites in the human host during a bite. Now, after that, the liver stage. Sporozoites travel to the liver, invading hepatocytes, the liver cells, and undergo asexual replication, forming schizones. So in species like Plasmodium vivax and Plasmodium ovale, some sporozoites can become dormant hypnozoites. Dormant hypnozoites meaning that they can remain there for around many years and will be active once they find a suitable time and a suitable place. So this one reason why these sporozoites they migrate to the liver, because the liver itself is a immunotolerant organ, meaning that it does not have the immune activity present in the liver. So that's the reason they migrate to the liver and go through the asexual replication phase. Next stage, the blood stage. Merozoites released from the liver enter the bloodstream and evade the red blood cells. So inside the red blood cells, the parasites undergo asexual multiplication, leading to the rupture of red blood cells, which releases the new mesozoites, causing symptoms such as fever, chills. Fourth, gametocyte formation. Some parasites differentiate into sexual forms called gametocytes, which are taken up by another mosquito during a meal. So the second mosquito will come and which will take in blood. So with the blood, it contains the gametocytes of the previous species of Plasmodium. So the gametocytes is taken up by the mosquito, and then in the mosquito's midgut, or we call it the intestine, gametocytes mature into gametes, fertilize to form zygotes, and develop into ookinetes. Ookinetes penetrate the midgut wall or the intestine and form oocysts. These oocysts, which release the new sporozoites, will migrate to the mosquito's salivary glands, hence completing the cycle, and the cycle will start once again. Clinical manifestations. So first is Plasmodium falciparum. So causes the most severe form of malaria with complications like cerebral malaria, severe anemia, and multi-organ failure. Then the Plasmodium vivax and Plasmodium ovale typically cause milder disease but can lead to relapses due to dormant liver stages or the hypnozoites. So these Plasmodium vivax and Plasmodium ovale, they are like dormant species. So they will remain there until they find a suitable time to become reactive once again. And lastly is Plasmodium malariae, causes the chronic infections that can persist for years, leading to nephrotic syndrome. That's the syndrome of the brain. Sorry, not the brain, the kidney, sorry. My apologies. And this is the life cycle of the Plasmodium species. So it is same as the pathogenesis, but here you'll get the diagrams to understand even better on each points that I have explained. Now let's look at the laboratory diagnosis. So first is microscopy. So game sustained blood smear. This is golden standard for malaria diagnosis. There are two types, thick smear and thin smear. Thick smear allows detection of parasites by concentrating the red blood cells. Because we know that these species, they multiply asexually inside the red blood cells. So that's why the thick smear is used, right? Okay, so that it allows the detection of parasites in the concentrated red blood cells. Then we have thin smear, helps in a species identification by examining the morphology, their structure, the size, the shape, about how they look inside the red blood cells. This examination will differentiate between Plasmodium species based on the size, shape, and appearance of the parasites, the morphology. Now the rapid diagnostic tests. These tests detect specific antigens produced by Plasmodium species using immunographic methods. So commonly used methods is Plasmodium falciparum histidine-rich protein 2. and Plasmodium lactate dehydrogenase. These tests are useful in field study due to the ease of use and quick results. Third is molecular methods. PCR, highly sensitive and specific for detecting and differentiating Plasmodium species. Second is quantitative PCR or qPCR, allows for quantification of parasite load. Then we look at the serological tests, the ELISA method. Okay, the enzyme-linked immunosorbent assay. This detects the antibodies against the Plasmodium species, which indicates whether they have a chronic infection or any other past infections related. Then indirect fluorescent antibody test, IFA, used in some clinical settings, but less commonly used due to their technical complexity and of course the cost. Now, let's look at some of the preventions. Mosquito control. So you can use, the individuals can use insecticide-treated bed nets. Sleeping under the bed nets treated with insecticides can significantly reduce the mosquito bites. The indoor residual sprays, okay, the spraying insecticides on the interior walls of homes can kill mosquitoes that rest on these surfaces. And it also reduces their mosquito breeding sites. So another thing we can do is stagnant draining, like clearing of blocked drains and constituents, and also using larvicides. Larvicides is like a specifically insecticide designed for controlling the larvae of the mosquitoes. Yes, in order to control the mosquito populations. Second is personal protections using repellents, containing DEET, picaridine, and other effective compounds on exposed skin and clothing can help deter mosquito bites. Personal protection, the clothing we use, the clothing, wearing long sleeve shirts, long pants, especially during mosquito activity times, such as the dusk and the dawn, because that's the period where the mosquitoes, they come. Screening, installing window and door screens to keep mosquitoes out of living spaces. Third, community and public awareness measurements. So educational awareness, raising awareness about malaria prevention and symptoms can help the communities to take proactive measures. With considering the health infrastructure, so strengthening the healthcare systems to ensure prompt diagnosis and treatment of malaria cases can reduce the transmission of the Plasmodium species. Let's look at some of these treatments. So for different species, there are different treatments. So these treatments are specific for each species. So first is Plasmodium falciparum. The first line of treatment is artemisinin-based combination therapies, the ACTs, such as artemether-lumefantrine and artesunate mefloquine. So these are the treatments used against the Plasmodium falciparum. Second is severe Plasmodium falciparum malaria. The first line of treatment is intravenous artesunate. The alternative treatments includes the intravenous quinine or quinidine, where artesunate is not available. So where this artesunate is not available, so these are the alternative treatments. You can use the intravenous quinine or quinidine, which is readily available, and chloroquine is typically effective for Plasmodium malaria infections. So if there's like a infection, then you can use the chloroquine. But if you're considering the treatments, treatments of Plasmodium malaria, then intravenous quinine is used. Last is Plasmodium vivax and Plasmodium ovale, since they use the same method of treatment. So chloroquine sensitive regions. So chloroquine followed by primaquine to eradicate the liver hypnozoites and prevent relapse. So this will kill the spores in the liver, which have been living, which have been dormant, to prevent the relapse. And the chloroquine resistant regions, the ACT, followed by primaquine. And this is it. This is the references. So I would like to thank you all for such a wonderful evening with me. And please, we'll be uploading many videos about many different parasites. So I would like you guys to subscribe and share this. And yeah, goodbye. Have a blessed evening. Thank you.